You said, I'm Ryan. I'm going to talk you through cystic fibrosis in the gut, and specifically how we sort of look at the organization and the function of the gut microbiota, so all the microbes that live in the gut inside of us. So a quick bit about me. I did my undergraduate degree at the University of Sheffield, and that's where I first came in contact with cystic fibrosis. So I was looking at more of a pharmacology sort of based project there, where I was looking at ways to sort of activate this. You'll understand more in a second, but the protein channel that's responsible for the disease, how can we activate it with drugs to help patients? From there, I did a master's at the University of Sheffield, and I did molecular medicine. And that's where I learned a lot of the techniques in theory that I apply now in my PhD. And again, I'll walk you through some of those. And during that degree, I specialized in microbes and infection towards the end of it, which leads me nicely to where I am now, really, doing my PhD in clinical microbiology, looking at the gut microbiota in cystic fibrosis. So it all tied together quite well. So what is cystic fibrosis? So if you didn't know, cystic fibrosis is the most common life-threatening recessive disorder caused by mutations in the cystic fibrosis transmembrane regulator gene. There's over 10,000 patients in the UK alone. And basically what it means is the recessiveness means that you inherit a faulty copy of the gene from your mother and your father, and if you have two faulty copies, then you have the disease. There's thousands of mutations, some are worse than others, but yeah. So in case you're not familiar, the sort of central dogma of sort of DNA, RNA, and protein is that we have DNA. DNA then becomes RNA via transcription, and it's that RNA that is then taken up by the cell's ribosomes and turned into these proteins that have all these functions inside each cell in the body. Obviously, all the proteins are different, they have different functions, and the cystic fibrosis transmembrane regulator gene encodes the CFTR protein, and that is one of these guys here. It's a channel protein, and it's a channel protein that's found at the epithelial cell surface. Now, what that means is anywhere in your body where you've got a tube system that has mucus overlying it, so in your lungs, in your intestines, in your kidney, in your pancreas, in your liver, that's where this protein would normally be expressed. So obviously, if the protein isn't expressed or isn't doing a very good job of like being expressed, or it's faulty, then you have an issue. And what that issue is that the protein normally lets chloride ions pass through it, which are negative ions. And what happens then is sodium ions would follow because they're positive. And then after the sodium would follow, you'd get water because water is attracted to sodium. So if you've not got any chloride moving through the cell, you've got no sodium, so you've got no water. So that mucus that's above those cells and the epithelium, there's no water in it. It's really thick and viscous. And that's kind of the hallmark of CF, really. So if you have heard of CF, you may be aware of the patients that have sort of recurrent lung infections and they have lots of physiotherapy and they're always coughing up mucus. So that's why, basically. So yeah, in brief, faulty channel protein, poor ion transport, thick viscous mucus over the epithelium. So what parts of the body are affected by CF? So there's a myriad of sites that are affected. As I said, anywhere where this protein is expressed in the epithelium, you've got an issue. So like I said, the lungs, where we have repeated bacterial infections and a decline in lung function, which unfortunately is the major killer in the population. Again, we have the pancreas, where these pancreatic ducts that are really important for secreting those enzymes into your gut, they get blocked with the mucus. So when those enzymes are released, they all just accumulate and they basically digest and inflame the pancreas to the point where it stops working. We have the liver, same sort of thing there, the bile ducts. So bile is secreted by the liver to help you metabolize and absorb fat, really. That gets blocked, so that's not doing a very good job. And we have the intestinal tract, which is my sort of focus, where we get blockages, inflammation, and obviously malabsorption of nutrients. And then just to add as well, we do have the brain indirectly, because as you can imagine, these patients that have this disease, it would really get you down having all these ailments and issues across your whole body. So there are high rates of anxiety and depression in the community, which is an issue. But in terms of the intestinal tract, I'll walk you through that in a second. But before I jump into that, I just want to get a few definitions out of the way. These words will pop up a few times, so just to get everyone on board. So the microbiota is a fancy term basically for all the microorganisms that live in or on the human body. So if I was to say the lung microbiota, it's all the microbes in the lung, the gut microbiota, all the microbes in the gut, so on and so forth. And there's 100 million microbes in the gut. And I think it works out that there's a 1-to-1 ratio with actual cells, bacterial cells in the gut, and the whole total cell count of the human body. So I like to think of us as like a superorganism. Obviously we are who we are, but we have a whole another world going on inside of us that can control our emotions, our gut health, other inflammatory immune related diseases. So it's really mad to think about. We then have the microbiome, which is essentially the same thing, but it's not just who's there and what bugs are there or bacteria. It's what can they do? So what DNA do they have? And what genomic content do they have? And what functions do they have? What metabolites can they make? What toxins do they secrete? So it's not just who's there, but it's what they're doing as well. And then the final term is dysbiosis. So if I was to say there is a dysbiosis in the gut microbiota, that essentially means there's a disruption to the community. So for whatever reason, that community's changed. And a resultant of that change, who's there is obviously different. And the functions they can perform are different too. So yeah, just bear those terms in mind because I will say them quite a few times after this. But if you go back to the intestinal focus of CF. So as I said before, we have intestinal blockages, and this can happen in the womb actually. So in babies, in prenatal, before they're even born, they can have a blockage in their intestine. I'm just going to show you a cross section of that now. So it's not too grim, but it is a cross section of the intestine. So this is it here, and we call this meconium ileus. So the very first poo that a baby would have or pass is called the meconium, and essentially what it is, it's all the nutrients taken from the mother, and it's that in combination with the mucus that's really thick and sticky in the intestine because of the disease. It creates a plug that blocks the intestine, and depending on what mutation the children have, it can be anywhere as much as 20% of kids are born with this. And if it isn't like flushed out with laxatives or surgically operated, it can be lethal and unfortunately kill the children. So it is really serious. We have intestinal inflammation, which can lead to further intestinal injury and increase the risk of cancer. And of course, we have microbial dysbiosis, which is what I'm going to talk about more in a second. And the important thing is that we think that microbial dysbiosis will feed back into this inflammation. So the fact that there is a dysbiosis, we think that will increase inflammation, increase the risk of cancer, and also increase the risk of blockages and other abnormalities in the intestine. So it's no surprise that these patients have loads of abdominal symptoms like pain, flatulence, bloating, constipation. They don't want to eat because every time they eat, it's painful for them. And actually, how can we relieve the GI symptoms in people with CF is one of the top research questions that was voted by patients, their carers, and the clinicians and doctors that care for them. So this is quite a big thing at the minute in the field. So yeah, I'm going to dive into the microbial dysbiosis a bit more and why the gut microbiota are important. But here are some important roles that they normally fulfill inside the gut. So the first thing is immune maturation. So the very first microbes or bacteria that colonize your gut basically help teach your immune system in your gut that it doesn't need to start going and attacking every single microbe it discovers. Because if it did, it'd have a real big problem because as I said before, there's 100 million microbes in the gut. If every one of those set off a signaling pathway in your body that was like, oh, we need to fight this, you just have constant inflammation and immune reactions going on. So these early bugs that get in there, they sort of train your immune system that not everything is bad that's inside of you, every microbe. Also, the early colonizers are generally good symbiotic bacteria, so they help us and we help them by giving them an environment to live in. And by them just existing and living there, that reduces the chance of pathogenic or bad bacteria having a chance to grow because they'll be competing for nutrients in that same environment. So it really helps us there as they keep out the bad guys that can really cause issues. We have the maintenance of gut barrier integrity. So again, these early colonizers and these good bacteria, they produce a lot of important metabolites. Some of them act as direct energy sources for the colonocytes, which is a fancy word for the cells that line your epithelium, your barrier in your gut. So these bacteria can produce things that our gut cells need to keep fit and healthy and strong, and to keep that barrier nice and strong and tight, so no pathogens can seep through the leaky gut. And then finally, we have the anti-inflammatory properties. Again, some bacteria secrete or produce short-chain fatty acids when they metabolize fiber. So when you hear the term, oh, eat a lot of fiber, it's good for you. By eating a lot of fiber, we can't digest it, or some of it. So the bacteria in your gut can. And it's those gut bacteria that digest that fiber and make these really beneficial products that help us regulate the intestinal inflammation and really keep everything under wraps down in the gut. So these are the normal roles that are fulfilled in the gut by the gut microbiota. And we do see that this is all disrupted really in CF. So this dysbiosis, it can happen in three main ways. So as I said before, this changes to the mucosal environment. So the fact that there's more mucus in the gut, that's better for other bacteria to grow in than the quote unquote healthy bacteria. So that the environment's physically different. And as I said earlier as well, the pancreas is destroyed. So those enzymes that the pancreas secretes to help us digest food, they just aren't there. I mean, there are replacement therapies where we do give them patients synthetic enzymes, but it's not as effective as the real thing, obviously. And what you find is all this leftover food, unfortunately, again, it's used by the bad bacteria. They just have a better tendency to use it as an energy source. So they expand and grow in numbers and start competing with the good guys that are doing all these important functions. We have treatment approaches. That's a real big driver of dysbiosis. The main one being antibiotics. So again, as I was saying earlier, all these patients with CF, they have a lot of lung infections. And the way that we used to treat those historically is just give them loads of antibiotics to target and fight the infection off. Now, the issue with that is, if you give them the antibiotic for the lung infection, the knock-on effect of that is it unfortunately wipes out the bacteria in the gut, some of which will be good, which leaves a massive gap open in that population for the bad guys, again, to just come in, swarm, and expand in numbers. And then below that, we have probiotics and CFTR modulators. Now, they've got an asterisk next to them because we actually think that they will reduce or reverse the dysbiosis. So you might have seen things in the supermarket like Yakult with probiotic drinks. It's not the best example. There are better ones out there, but it's a very popular one. And essentially, that's us, or products where we give patients drinks or food that contain millions and millions of colonies of good bacteria in the hopes that they reach the gut, they seed, and they expand in their colonies and they fight off or compete with the bad guys. And then we have CFTR modulators, which I think Helen will talk to you a bit more about. And they're fancy new drugs that, if you remember that first or second slide I showed you, where I showed you about the DNA and the protein made and the faulty protein doesn't work and the mucus changes because of that. Well, these CFTR modulator drugs fix that dodgy protein at the start. So the hope is that if we fix it at the start, the protein, it'll sort the mucus out. And by sorting the mucus out, we should hopefully revert those bacterial communities back to the healthy, quote unquote, healthy composition. And then we have lifestyle. So classically, patients with CF, if they have a high energy diet, then they do better with their lung function. So what was recommended is a high fat diet, because fat is the highest, I think 1 gram of fat is more calories than 1 gram of protein and 1 gram of carbs. So a high fat diet was recommended for patients to maintain their energy requirements. And unfortunately, again, by having high fat, the bad bacteria are really good at metabolizing and using that fat to grow. And I've already said before that the pancreas isn't working very well and the liver's not working very well. So again, we can't metabolize that fat very well. So it kind of exacerbates the problem. By trying to fix one problem, you sort of create another problem. And then we have reduced physical activity. Obviously, these patients have lung infections. They're not very physically fit. Their cardiovascular health is not very good. That will impact the microbiota. And as patients get older, their microbiome changes as well, naturally. So these things all play into it. So we know there is a dysbiosis, but how do we know who actually is there? So in an ideal world, okay, so the first problem is, how do we actually sample the gut microbiota? I mean, if everything was simple and people didn't feel pain, we could literally go into the intestine and we could take a snip of the intestinal tract with the mucus and the epithelium and take a biopsy and then analyze that biopsy in the lab. The issue of that is it's very invasive. As you can imagine, it's not very nice having a biopsy in your intestines. Plus, if we want to compare the composition of the bacteria to healthy control subjects, you're going to find it very difficult to convince a lot of healthy people to go and have some of their intestines chopped out. So that's a real issue. And then on top of that, you'd also need a very trained specialist or clinician to actually perform the biopsy. So there are a few issues that we would have. So the solution to overcome that, you may know, you may not know, is actually using stool samples. So it's not invasive. It's just a little gross, obviously. But there are millions and millions of bacteria per gram of stool, which is quite disgusting when you think about it. But it's great in terms of the capability we have to use that to see what's going on further up the intestinal tract. And obviously it's much cheaper and easier to sample than a biopsy. So once we have that stool, how do we know like who is there? How do we actually determine which bacteria are present? And if you think about bacteria and looking at bacteria, you may be sort of familiar with the agar plate technique, where you get a single colony of bacteria and you streak it out, or you streak out bacteria until you get a single colony, which is like the little dots. Now, the problem with that is we can't represent those millions and millions of bacteria on a single agar plate. And bacteria, believe it or not, are very fussy. If we have one agar plate with a set of nutrients on it, that won't accommodate the whole community. Some bacteria will prefer different substrates to grow on than others. So there's not really a one shoe fits all approach for agar plates. And plus, most of the bacteria that inhabit the gut, especially the colon, are anaerobic. So they can't even grow in the presence of oxygen. So all you'd find on that agar plate is just all your aerobic bacteria, which obviously would be very biased, and it wouldn't represent a good snapshot of the community that you're looking at. So what's the solution to that? Well, we know that in that poo, there is all the bacteria we want to see, or we want to understand that are there. And we know they all have DNA. So if we can sequence or DNA sequence a certain region of that DNA, then we can compare it to a database and we can see which bacteria are there using really fancy high throughput computers and computers and sequencing programs and machines, which Helen's going to talk you through a bit more how that all works. I'm just going to gloss over it here. So which part of the DNA do we sequence to identify the bacteria? So I want you to think about a garage, a huge warehouse full of cars, and relate that to a bacterial population in the gut. So every car is a different bacteria, so on and so forth. Well, if we wanted to see, well, every car, we basically want to look for something in the bacteria that every bacteria has. There's no good comparing something if one bacteria doesn't have the same protein as another bacteria, because then you need something that everything has. So if you think about a car, every car has an engine. So we could look at every car's engine and say, yep, that's probably this car. Yep, that engine's this car, so on and so forth. And likewise, every cell has a ribosome. Ribosomes are super important for making proteins. Without a ribosome, you have no proteins, you have no functional cell, really. So we can use the ribosome as a marker. And just like we can look at a blueprint for the design of the engine, we can look at the blueprint for the design of the ribosome, the DNA, the ribosomal DNA. So essentially, we can use that ribosomal DNA, and there's a few intermediate steps that happen, but the sort of short of it is we look at the whole ribosomal DNA from the community and we sequence it on a fancy computer, and then we compare it to a database, and then it sort of tells you, oh yeah, this matches with this bacteria and this matches with this. And that's like the short answer of how we do it. So what can we do to investigate what the bacteria are doing? So i.e. their function, not just who's there, but what are they doing? And there's two approaches for this. There's a predictive analysis. So in that poo, as I said, there's all the DNA from the bacteria. And there's not just the ribosomal DNA. There's there's just the entire DNA, the whole genome of every bacteria is in there. So we can use even more powerful and stronger bioinformatic computers and techniques to look at all that genomic content at once. And we can map it to databases and work out which proteins the genes code for and what the proteins do. And we call that meta-genomics. So meta meaning like the current. So what I've just shown there would be the metagenome of the stool sample. I could go to the soil, I could take some soil and extract all the DNA. That'd be the metagenome of the soil. So that's, meta means current, and genomics, we're looking at the current genomic content of the sample, which is stool in this case. But we can also look at the direct analysis of the metabolites. So obviously those bacteria, they produce things. They produce important metabolites. Some of them produce toxins, which are dangerous. But the idea is those toxins and chemicals and metabolites will also be in that stool sample. So we can directly measure them with fancy chemistry analytical techniques like gas chromatography, mass spectrometry, or on the right, we have nuclear magnetic resonance spectroscopy, really fancy chemistry techniques. Well, they sound fancy actually, but they're not too complicated once you've used them a bit. And we call this process meta, looking at the current environment, bolomics, so the metabolites in the current environment, metabolomics. So if we bring all of that together, if we want to understand the organization and the function of the gut microbiota in cystic fibrosis, then we can take our clinical patient data, so the antibiotics they're taking, the inflammation they have, the diet, their age, their BMI, their gut function, their CFTR status, what mutation do they have, how much function of the protein, of the CFTR protein do they have. We can combine it with the community characterization, so that ribosomal sequencing I was mentioning earlier. We can then throw in the metagenomics to see what could they be doing, what's the predicted functional capacity. And then we can throw in the metabolomics, like, oh, what are they actually making? And yeah, if we combine that all together, that gives us a nice comprehensive view of what's going on in cystic fibrosis in the gut microbiota. But if you take a step back and just forget cystic fibrosis, the point of this is that we can apply these same techniques to any disease that's associated with the microbiota. That could be neurological diseases, cardiovascular diseases, cancers, and we can even step away from diseases in humans. We could go to a bunch of soil and say, these plants aren't growing very well. Let's see if they have a different composition of bacteria in the soil. And let's see if that bacteria in the soil is making different things that are affecting the plants. So the take-home message really is that the techniques that we use in this lab can really be applied to any walk of like microbial sort of biology, where you want to sequence and understand how communities are functioning and acting together. So what do we know so far? So we have got a paper out so far, and that is essentially where we looked at healthy control patients compared to cystic fibrosis patients, and we showed that the microbiota is significantly different between the CF and the healthies. We also showed that those important bacteria that produce those beneficial products earlier, they are massively downregulated in the CF population. We then showed that the lung antibiotics that the patients are given really drives the differences seen, has a massive effect. It actually had a bigger effect than the disease itself, which is substantially, it was pretty crazy when we found that. It was like, okay, the antibiotics really do mess things up. And then finally, we also showed that the actual physiology of the gut. So we did some fancy MRI scanning of the gut, and we showed that if you compare that data with the actual bacterial composition data, the patients that have slower transit of food because their gut's more thick and sticky and there's more mucus, they have different bacteria too. So we've shown that all these different parameters in disease do relate to the bacterial composition. And the future work we're going to do is looking at those CFTR modulators and those probiotics that I mentioned earlier. So that's a wrap anyway for the gut side of things, and that's just an overview of the things we do. If you've got any questions about any of the techniques, CF as a disease, the microbiota, or any general science career chat, potential undergraduate degrees, how did I get from here to here? What was my decision making? Drop me an email. More than happy for you to do that. And then I've got some QR codes there, which should take you to the pages above. So you've got the Cystic Fibrosis Trust, which is the charity that actually supports my PhD work. Obviously, it's a great charity. We have Guts UK, which is a more generic guts charity looking at not just cystic fibrosis, but bowel disease. And then we have the MMU page for the Bioscience Centre, so you can see the other research that we've got going on. But yeah, that's a wrap from me. I'll stop sharing. I'll hand over to Helen. And yeah, thanks for listening.
Okay, see if I can do the present a window. I think that might be the right one. I think it's this. Hopefully that comes up. Right. Does that look okay, Ryan? Can you just tell me if that looks...
Yeah, no, that's fine.
Oh, you've got a question there. Answer that question first while it's still fresh in everyone's head.
Let's have a look. So is it possible to isolate the different species of bacteria in the gut from stool samples? So in theory, it is. As I said before, in the gut, actually what happens in the gut is, as you start at the top of the intestinal tract, there's obviously more oxygen. As you work your way through the small intestine, get to the colon, large intestine, that ratio of oxygen just depletes to nothing. So in theory, if you wanted to see the bacteria, you could have really fancy, expensive, and we call them anaerobic chambers. So you can culture bacteria on agar plates, but it's inside a chamber that there's no air in it. So the bacteria can grow. But obviously, when there's millions and millions of bacteria, this is just not a plausible thing to do. You'd probably spend a year just streaking them all out on a plate, and you'd have to wait days for them to grow, and it's just a mess. But the beauty of the ribosomal sequencing is you can, depending on what region of this DNA you look at, you can get a species level identification. So you can actually, it's really, really good, and it's really, really specific. There's obviously limitations here and there, but it's much faster. We call it high throughput sequencing because we look at loads of data at once. And yeah, we just need fancy computers for it and DNA extraction methods, no fancy culture techniques. I'll answer these two questions. Yeah.
Oh,
that's fine, yeah.
All right, okay.
Go on then, I'll pass on to you, Helen. Yeah. Yeah, that's fine, yeah. I can answer those later. Go on, Helen.
All right, cool. And it gives you a chance, Ryan, to think of a good answer. Right, okie dokie. Hello, everybody. I hope you're good. My name's Helen, and I'm going to be taking you through a brief overview of the lung microbiota and cystic fibrosis. We're going to have a discussion about some of the techniques that we use regularly in the lab, the implications on patients, and the future of cystic fibrosis as well. So yes, I thought I'd just do a quick little introduction again. So I've taken a kind of a long way around to where I am now. I actually started in forensics. So I started my undergraduate degree in forensic biology, learning how to detect blood samples and all kinds of stuff from crime scenes. And then I took a bit of a swerve and I decided to look into medical sciences. So I took my master's degree in medicine instead. I then got my first job as a research technician at Newcastle University, which was a really, really interesting job. I got to help set up a organ retrieval unit at Newcastle at the NHS building there. So I've had the opportunity to receive and process donor organs from people, and we process them for use in research at the university. And I was also very fortunate to process an organ that was then actually put into another person and save their life, which was very, very cool. I then decided to take a PhD, I've just completed that a couple of months ago. And that got me into the world of cystic fibrosis. And I'm now the Senior Research Associate at the Manchester Metropolitan University. And we are looking at the bacteria and the fungus in cystic fibrosis in the lung. And we're also having a look at the impact of new medication in cystic fibrosis and what it does for the patients. So I've got a nice snazzy picture of me and my hairnet just before we got all sterile, ready for taking on an organ. We had to do the whole kit and caboodle, hairnets, really long gloves all the way up to the shoulders. It was a look, I'll tell you that. And I also do a bit of baking in my spare time. So let's just crack on with it, I guess. What is the lung microbiota, and what is the difference between a healthy lung and a cystic fibrosis lung? So Ryan's touched on this a little bit, so I'm just going to give like a really rapid overview of this. But basically, the lungs were once thought to be sterile. And when I say once thought, I mean like 20 years ago. It's only recently that we've come to realize that actually there's things living in there. And that is a really big community of bacteria, viruses, and fungi. Even in the healthy people, you will have something in your lung. And that community, as Ryan pointed out, is called the microbiota. So every time you breathe in, you pull in millions and millions of bacterial particles into your lungs, alongside thousands of fungal spores and viral particles. Finished my cup of tea, my mouth's gone all dry. So basically, If you are healthy, your lungs will be very, very good at removing all these particles from the lungs, and that prevents disease because these particles don't have a chance to settle, get familiar, and then cause disease. However, people with lung diseases, their lungs don't function as well, and as such, they won't be able to remove these particles, which gives those particles a really good opportunity to start creating an infection. So this is a super zoomed in scanning electron microscope image of a healthy lung. So all these little finger projections you see here are called cilia, and these are usually coated with a thin layer of nice sloppy mucus. So anything you breathe in is captured by the mucus, and then the cilia waft it all up your lung into the back of your throat, and then that's either coughed up, which is phlegm, or it's moved down into the stomach for disposal. So most of the phlegm and snot that you create in a day, you actually swallow and eat, which I always think is a really grim but really cool fact. So what do these cilia actually do? I actually managed to find a really nice little video here. So this is a culture of lung cells in a lab on a little plate, and there we go. Hopefully that comes through. You can see the little fingers wafting around, doing the little thing. So if there was a layer of mucus, that would slowly move the mucus across the surface and up and out of where it needs to be. And that's happening in your lungs all the time. It is. Okay. Oh, there we go. So the cystic fibrosis lung, completely different story. So cystic fibrosis patients go through a constant vicious cycle of infection, inflammation, scarring, and then reinfection. And this has a really devastating effect on the lungs. So obviously you can see here, we've got these patches where you can see all of the cilia are all stuck together because the thick, sticky mucus is really weighing them down, making it difficult for it to be removed. You've got patches where there's no cilia at all. And of course, this can also lead to something called a compound cilia, which is where several of them grow into a clump and then they can't move at all, which also contributes towards not being able to clear the lungs out. So the questions we need to ask are, who are living there in the lung in this thick mucus, and how do we identify them? So these are some of the main players when it comes to bacteria in the CF lung. These are the big six that we tend to look for in the clinic and in the hospital. Patients will go in every couple of months, provide a sample, and what we'll do is we'll streak all of this out on an agar plate, which you can see all along the bottom here. Oh, my mouse is showing up good. And then we'll try and figure out who is who. Now, as you can see, when you look at all the agar plates, they all look kind of the same. It's just a sort of white smear on some agar. And also these electron microscope images at the top, again, they all kind of look the same, you know, with the exception of Staphylococcus aureus. They're all these kind of rod-shaped kind of little beans. So how the hell do we figure out which one is which? Similarly, we also have fungus in the cystic fibrosis lung. Here's a couple of examples of the different ones that we come across. Unlike bacteria, fungus can take on two different forms. So you can see here we've got Candida albicans and we've got Malassezia restrictor at the end here. These are the yeast form of fungus. And then in the middle here, Aspergillus, Exophiala, and Pseudosporium take on a mold form. So that does help when it comes to plating them up. You can see they're all different colors and they've all got slightly different formations, and that does help. And under the microscope, they also have slightly different formations as well. However, these patterns will be repeated across loads of different species. So we still have to narrow it down and figure out who is who. So in the hospital, they use something called culture-based detection. So basically what happens is the patient will come in, they will provide a sample. So that will be, they'll either cough up some phlegm, some sputum, or we'll take a swab. So we'll either swab right at the back of the throat, or we'll get a swab, we'll put it into the mouth and they'll cough onto the swab. And then what we'll do is we'll smear that on some agar and we'll see what grows. And then those growths are then tested further to determine what they are. We have various techniques to kind of narrow down who is who. So the example I've got here is catalase testing. So both Staph aureus and Pseudomonas are catalase positive. And what this means is that we will pick up a little growth and we will put this liquid on it. If it froths up, it's catalase positive. If it doesn't froth, it's catalase negative. And there's a whole catalogue of different detections that we can use to narrow down who is who. Now, while this is the gold standard in hospitals and how we've been routinely checking cultures for quite a long time now, it does have its limitations. We can get things like false positives and false negatives. Bacteria in particular are really good at sharing their genetics, and some bacteria will acquire the ability to not be catalase positive or become catalase positive, which can really screw around with the results that we get. So that could be a false positive or generate a false negative. Some pathogens just simply will not culture. We can smear them on as many different agars as we want, and they just don't grow. And this is sometimes caused by a phenomenon called viable but not cultural, or VBNC. And this is a protective mechanism that the bacteria use to stop themselves from being hurt by antibiotic treatments. So what will sometimes happen is a patient will come in, they'll give a sample, we'll find they're positive for something, we'll go, right, okay, we'll get some antibiotics on that. They'll come back a couple of weeks later for another sample, and the sample will be clean, and we'll go, oh, great, the antibiotics worked. But in actual fact, the pathogens just aren't culturing because they're trying to protect themselves from the antibiotics we've just bombarded with them with. So we think that the patient's been cured when in actual fact they haven't, which is, of course, a big issue. And then, of course, we have the fact that hospitals may only use a certain range of different agars to grow things on. There are guidelines and there are recommendations by the NHS, and I don't know if you've heard of NICE, they're the big body that oversees medical decisions in the UK. And most hospitals will follow like the basic thing of that. Some hospitals might decide to do a little extra. Some hospitals may have found something in the past and decided, you know what, we're going to use this specific agar as well. And that just means that each hospital has a different range of tests and a different range of agars and a different range of results that come out of them, which can make collaborating data really, really difficult. So then what the hell do I do? Well, basically, I have several questions that I need to answer, and that is, who is in the lung? Does this change over time? So we take samples over several years and we compare the fluctuations over time in patients. Do antibiotics have an impact on who is living there? And we have a lot of new medication coming out lately, and they've all been really wonderful. And we need to know in the long run, how does this impact patients? Is it a really good short-term change and then it sort of calms down? Or is it a really good impact that continues for a long, long time? We don't know. And that's the things that we need to find out. What does that look like? Well, simply put, I have an entire freezer of other people's phlegm in the lab, which is a lovely icebreaker to any conversation. And it looks a little bit like this, these little tubes of phlegm. So what we do is we process the samples, we put them through a kit that gradually cleans the DNA out of the cells. And then what we do is we sequence that DNA to figure out who it is, process that, analyze the data, and then compare it to things like how old the patients are, what their lung function is like, what antibiotics have they been on, things like that, to see if there's any patterns in that data. I've put down here a little snapshot of a previous study that I did for my PhD, which is looking at all the different fungus in different patients. So each of those columns is a different patient. And as you can see, each patient has a really vastly unique fungal makeup. So no patient is the same. And that means that there isn't one size fits all treatment when it comes to different people's infections and how those infections interact with the other microbes in the lung. And that brings along, you know, do we need to start thinking about things like personalized medicine and all of that sort of stuff? And this is stuff that we really need to research so that we can really improve treatments. So what I use, which Ryan has touched upon, is molecular-based. Oh, sorry. Yeah. Oh. Okie dokie. Oh, right. Okie dokie. Yeah, of course we can. Yeah. That seems to have worked. Everyone seems to have moved over. Oh.
Yeah, I was conscious about that, so I started like typing my questions out.
I didn't realise it had a time limit. How rude. How rude. Well, that's no good. What if you've got someone who really likes to talk like me? Honestly. Right, I will start sharing my screen again. Like this. Hopefully that comes up. Yep, that looks all right. Is that everybody? I think that's everybody. Yeah, okie dokie. So, as I was saying before, Google very rudely interrupted us. Ryan has touched upon how we seek DNA in a specific sequence. So in order to find out, for example, bacteria, what we're looking for, we will find a little section of that sequence that every bacteria shares, okay? And then everything after that will be completely unique to that specific bacteria. So what we do is we use a primer, which is a little target region. We attach it to this shared piece of DNA, and we get a machine to basically go along and sequence the rest of the DNA after that. Then what we can do is we can compare that sequence to a massive database to figure out who we're looking at. Oh, I've not clicked on the, there we go. So this is what it kind of looks like. And this is the thing I find really, really cool. So for my PhD, I used a sequencing platform called Illumina. Now, the image on the right here is the flow cell, and this is where all of the DNA sequencing takes place. That chip is about half the size of a credit card. It's tiny. And on that chip is a really, really, really thin piece of glass. Okay. And what we do is we put our DNA on the glass and it attaches and it all sticks up in little strands, like on this image down here. So what we do is we take one of the bases that make up DNA and we flood the glass and the DNA with one of those bases. If the base is the next one in the sequence, it attaches and it lights up. Okay. And the machine takes a photo of this and records the data. Okay. Really simple premise. But the really cool part is that this is a picture of the DNA. So every single one of those lit up dots is a piece of DNA with a base attaching to it and lighting up. I just think that's really, really cool because like you can literally watch DNA being sequenced in real time through little photos like this. And technology is just, it's just great. I think it's really neat. So how has all of this research helped fix cystic fibrosis? So basically, 70 years ago, if you were born with this disease, you weren't expected to reach 10 years old. You'd be lucky if you got to 5, which is absolutely horrific. It is. Nowadays, we're looking at people living into their 50s and 60s, which is absolutely phenomenal. Infection rates have dramatically reduced across all patients because we've realized that antibiotics can really help stop that vicious cycle of infection and inflammation. And we've also started to realize that infection is actually one of the biggest killers. In fact, it is the biggest killer in this disease. We have new medication that addresses the genetic part of the disease, and it's dramatically improved the quality of life for patients in recent years. And of course, because of sequencing and all of this, we're slowly beginning to understand the roles that these microbes play in disease. We have several pathogens now that we realize are actually helping protect the lung. We have a couple that we're not too sure about. We have evidence to show that on the one hand, they stop bad bacteria from colonizing, but on the other hand, they also protect the bad bacteria. So we need to figure out whose side they're actually on. And all of this comes together to helping patients live better lives. And in extension to all of this, there are so many other lung diseases that we can apply all of this to. Asthma, COPD, bronchitis, they all have bacteria in their lungs. Those bacteria and fungus and viruses will be contributing in some way to the progression of the disease. But how? We don't know. And that's what this research is all about. So I mentioned that we have these really, really new drugs. The newest drug is Trikafta, which is on the bottom right there. It only got put out for general usage, I think, about a year ago, and it has revolutionized CF patients' lives. Usually, cystic fibrosis patients go to a hospital at least 5 times a year. They're riddled with antibiotic treatments. They have to constantly do physiotherapy to push the mucus up and out of their lungs. These people don't go to the hospital anymore. They barely even turn up to clinic anymore, and they don't cough anything up. We've basically kind of prevented all of the problems, bar a couple of things, with these new drugs, which is really, really good. Unfortunately, these drugs are only effective in a certain set of genetic mutations, and roughly about 70% of the population will be eligible for these kinds of drugs. But that means that there's still a significant amount of people who just aren't benefiting from these. But that, again, research will help us to identify ways of helping them. So how do these modulators work? Again, this is another really cool bit of technology in pharmaceuticals. So there are 2 types of drug within these medications. There are things called potentiators. So ivacaftor is a potentiator, and that helps the defective protein function correctly. It ensures that when it's been made, it's installed correctly in the cell wall and that it opens and closes correctly. And then on top of that, we can combine ivacaftor with a corrector like lumacaftor, which takes the raw protein as it's been made and it helps it fold correctly in the first place, so that when things like ivacaftor take over and install it into the cell barrier, it's much more likely to work properly. So stopping short of genetically modifying people with cystic fibrosis, we are effectively cancelling out the genetic mutation with these types of drugs, which is absolutely amazing. However, like I say, there are still a lot of issues that we need to address and we need to research. And it's stuff like that that myself and Ryan got into science for. And this is the reason why we do these kinds of things, so we can figure out how to help other people. So I have a few links. I'm assuming Abby will share these with you guys. So there are a couple of social media bits. So there's Ladies in Lab Coats, and they look at things like cell biology and Parkinson's disease. One of the girls actually works with little worms and creates crystals, which is actually really, really cool. We actually work with one of the Ladies in Lab Coats, which is quite nice. Soapbox Science is a company that promotes accessible science to the general public and also women in STEM. They do little pop-up events all around the country, so it might be worth checking them out to see if there's one near you. Of course, there's the Cystic Fibrosis Trust and Guts UK, which Ryan touched upon. I'm not sure if there's anything similar in Birmingham, but Manchester has a science festival every single year at the Science Industry Museum. There are all of the talks have been recorded and are actually online at the Science Industry Museum website. So if you're interested in having a look at some other areas of science or other bits of biology, there will be something there for you to have a look at. And of course, at the bottom there, I've made a link to the American Society of Microbiology, which talks about careers in microbiology, what kind of degrees and qualifications you need to get, ways into microbiology, and the type of work that you can expect in a microbiology career, if that is something you quite fancy doing. So with that, thank you very much for listening. I think we've got the Q&A session now. So I'll stop presenting, and then Abby, how do you want to do the Q&A session? I would say so at the moment, yes. Yeah, the drugs are getting a hell of a lot better. The current drug Trikafta is I think it's the 3rd generation of modulators. And I know that Manchester Hospital has actually signed up to trial the 4th generation. So we're constantly finding new drugs that will help to, modulate these proteins in the cell. However, these drugs do come with side effects. So the current drug that we use, Trikafta, it actually gets metabolized by the liver. So it gets broken down in the body by the liver, and it can accumulate there and cause lots of problems in some patients. So whilst it is a really, really good drug and really effective, there are still some drawbacks that we need to assess. And hopefully, who knows, some future generations of these drugs will not have as many side effects, and more people will be able to take them and be quite happy on them.
Just to add to that as well, the big thing as well is getting these drugs in children earlier. So I think the current licensing now is 2 years old, I believe, is as young as it can go now.
I think it's 6.
Oh, is it
6 to
6?
9.
Oh, okay,
Yeah,
6 to 9.
6 to 9-year-olds.
Obviously, a lot of the issues that happen in cystic fibrosis do happen in childhood. So if the pancreas is already destroyed by the time they're 6 years old, then the knock-on effects of that are still going to happen in the gut and the gut microbiota. So if we can get in earlier and modulate the protein from birth, basically, or even in the womb, that could be possible in the future. Obviously, the gold standard would be genetic editing, which is obviously, you can't really do that. So it's like this is the next best thing, isn't it? But yeah, I think the earlier you can get in, the better. And obviously, as time goes on, we learn more, and we have to understand the safety profile of new drugs when we test them. So that's the main thing, isn't it? I think moving forward as well.
Yeah.
Do you want me to take it, Helen? Or
I don't know about gut stuff.
Oh yeah, it's gut. Sorry. So are treatments more effective in cultivating a healthy gut microbiome? So yeah, essentially what we know is essentially the earlier you get in, the better. We know that if you compare the gut microbiota in healthy children to CF children, as the patients get older, the composition just keeps moving away. So if you can get in there at an earlier time point, then you're reducing the chance of that diversity being lost and all the functional consequences that come with it as well. And like we were saying, as time goes on, these modulators get licensed for younger and younger patients. And there's other things in childhood that influence the microbiota. I mean, if you're born via a C-section, you are predisposed to a different gut bacteria than if you were born through the uterus naturally. So there's diet, breastfeeding massively impacts the gut microbiota. So there's things that can be done on the side to modulate. Obviously, the main drivers of that dysbiosis are the mutation and the protein issue, which leads to the mucus, and obviously the antibiotics. But there are things you can control to try and alleviate the problem. Yeah.
Oh.
On,
So I'm very fortunate that I have a job that I absolutely love. For me, science is the best job for me because I'm not sat behind a computer for most of the day. I get to go off into the lab. I get to create things, destroy a lot of bacteria, play around with life essentially, which is really, really interesting. And I find the techniques that we do just really cool. Like I I get to find out what is living in a stranger's lung, and see if that can help make them better. I mean, that's what's interesting to me. I want to make medicine. I want to contribute towards making people feel better. As for studying cystic fibrosis, I kind of accidentally fell into it, but it's proven to be really challenging but really rewarding area of research, and it is challenging. But the thing is, I think as well, it's really important to emphasize that I know a lot of places seem to put biology and the sciences as something that you need to be a straight A student at, and you really don't. Like I did terribly in my A levels. I failed maths GCSE, and I'm still doing this job. You don't have to have necessarily the academic smarts to do good science. You know, that I've met a lot of people who are extremely clever, but they can't do anything in the lab. And if you can't do the physical lab work, well, you know, so yes, it can be challenging, and it can be really difficult, but at the same time, it is doable for everyone. So please, if someone says, "Oh, biology is for smart people only," don't listen to them. Just don't listen to them.
Yeah, I mean, quickly, for me, so when I was younger, I didn't really have anyone older than me and my family that had gone to university. And I did science and I enjoyed it. And I fell into that classical pathway that a lot of people do, where they're like, oh, I'm going to be a dentist, or I'm going to be a doctor, not really understanding the sort of breadth of science careers that are out there. Yeah, like Helen, I flunked out a little bit on my levels, didn't do too great. I thought, okay, what can I do? I really enjoy science. Did biomedical science, which I think is a good platform if you know you're interested in science, and you might want to do some research, but you might also want to do dentistry or medicine after. Obviously, biomed's quite a safe bet. So yeah, just went from there, and then like Helen, just fell into CF, and then thought this is pretty cool. Working in a lab's pretty cool. You know, you can have your music on sometimes. You sat away doing your work. It's not all stress, regimental working. So, and there's flexibility. So yeah, just the lifestyle's great.
Ooh, right. So basically, all of them overlap. We have shown that bacteria can attack each other and shield each other. We know that some fungus actually prevent other fungus from colonizing an area. It also helps keep other bacteria in check. And we also have some evidence to suggest that viruses can play a part in helping keep fungus and bacteria in check. The problem we have is that a lot of research only looks at this one thing here and this one thing here, and how do they interact, when in actual fact, that's not how it works in the body. You have billions of things all interacting at the same time, but it's really difficult to replicate that in the lab. So what we know is that some things might work together, some things might work against each other, and they may do that in different combinations, but we don't know what they all do as a group. And that's the key question that we need to answer.
Yeah. Yeah, I'll jump in, Helen.
Yeah.
I'd say I wouldn't say they cure it out and out, because lung functional won't really ever return to 100%. But they do really help, and they are the gold standard, if you like. Obviously, gold standard being the best that you can do. They are the gold standard approach, and like Helen said, they cover about 70-80% of the population, the ones that are out at the minute. And this is the sad sort of reality, is that there are people that have these really obscure mutations that these modulators just don't work for. And obviously, from a pharmaceutical perspective, it's hard to convince the big pharma companies that they need to invest millions and millions and millions and millions of pounds to just help 100 people or 1,000 people. So there's a gap that needs to be bridged between the community, the scientists, and the big pharma companies that are funding these sort of research programs. So there's a lot of politics in science. That's one thing you will learn. And it is sad. And that's why doing research and understanding how diet can impact it, antibiotics, probiotics, can just help these other people that can't have the modulators. So it's like a second best approach, which is obviously better than nothing. So, yeah.
Very hard. So we actually plug my paper here. We just released a new paper having a look at what is cultured in the hospital and what is actually in the sample. So we took patients over 3 years and we took all of their hospital results and we compared that to molecular detection of pathogens in those same samples. And we found that culture was not picking up nearly any of the actual pathogens that were in the samples. So the problem we have is that, first of all, what we are doing in hospitals with patients at the moment is picking up some things and it is helping to treat some infections. But the true scale of these infections are not actually being detected by the hospitals. Unfortunately, setting up molecular detection methods is expensive initially and it's time consuming and you need to train people. And this involves us trying to convince the NHS to really invest in this sort of stuff. And it's not even just for cystic fibrosis. Whenever you go to hospital and you get something tested, it gets sent off and it may actually end up being looked at in a molecular way. So it does happen. It's just, it doesn't happen often enough. And I think the next stage in really revolutionizing treatment across all different kinds of diseases is introducing these next generation, these brand new technologies into hospitals and using them regularly so that we can find these pathogens that are trying to hide from us and really addressing infection properly.
Mm-hmm. Yeah, so it 100% can. And I should have mentioned that actually. It's called distal intestinal obstruction syndrome. So it's basically like meconium ileus, but in adults or in older people. And again, it happens in around 10% of the population. And again, you need laxatives or you sometimes need operations to treat it. And it's, for example, there's evidence to show that patients that, I don't know why you wouldn't take your treatment, but patients that don't take their pancreatic enzyme replacement therapy are more likely to have it because there's a buildup of fats and mucus, and that's going to block your intestines. So yeah, the short answer, yes, and it is a problem. Yeah.
Both of those are yes. So there are some companies that do specialize in different types of science. You have oncology research for cancers and things. There is actually a couple of microbiome research companies up and down the UK. But research like what we do, I think, Ryan, correct me if I'm wrong, it's kind of university based. It's a lot of collaborations between different universities, getting lots of different funding from different places, and working with hospitals to collect samples. And then,
I think a generic thing would be so there's a big company called Vertex Pharmaceuticals. They make all the drugs really for CF, or they make the majority of them. They have all the money. They then divvy that out to the separate charities. And then the separate charities offer that money to universities or researchers like our lab head, our boss. He'll get the grant money and then he'll invest that into research, into paying for Helen, paying for me to do my PhD, and then we'll do the work. We'll pay for the consumables. So it filters down. So it's not, you don't work necessarily for the big pharma companies. There will be internal jobs, but I think most of it trickles down through academia. That's how it sort of works. Yeah. But yeah, I mean, of course you can work in sort of, and this is quite a niche disease. I mean, it's niche, but it's still the most common. recessive disorder. But I think for more like general lab work or general like microbe infection stuff, I think there's a lot of private firms out there. But yeah.
Plus, as well, there's positions like lab technicians and research technicians. So I worked as a research technician at Newcastle, which basically meant if there was a project and they needed some help, I was the person who went and helped on it, which means that you get exposed to doing lots of different projects, but you're not kind of tied down to a single subject, which can be really good for CV building if you want to build up a skill set of various different things. Mm-hmm.
I mean, it's not really my forte, but from what I understand, it's a bi-directional relationship. I mean, I think everyone's had it before where if you're nervous, your tummy might feel a bit, like there's evidence to suggest that, we know if we go from the gut to the brain, we know that some of the metabolites, for example, I think maybe it's either serotonin or dopamine. The majority of that is produced in the gut by the gut bacteria. Obviously, serotonin makes you happy, mood increaser. Again, dopamine sort of reward stimulant. So we know that it definitely works from the bacteria to the gut, from the gut bacteria to the brain. I'm not too sure about the reverse psychology. I mean, what we do see in some trials is obviously you might have heard of placebo effects. If people think they're getting a drug, sometimes it seems to work, even though it's a placebo. So you could argue that that is them mentally thinking and the gut bacteria is changing. So I guess that's evidence that it's bi-directional relationship. But yeah, not really my forte, but yeah, there's definitely evidence to suggest that the gut bacteria modulate mood. And they do say, don't they, if you eat a healthy diet and you work out and stuff, you generally feel better. So maybe it plays into that. I don't know.
So basically, we have managed to find that there are certain bacteria in the lung that actually prevent other bacteria from becoming aggressive, even the ones who have antibiotic resistance. So in effect, you essentially end up with a little war going on in your lungs without you even having to do anything in the first place. I think is it staph? I think it's staph. It kind of like finds Pseudomonas, sits on it, and then eats it, which is really cool. But it stops Pseudomonas from getting infections. Plus as well, we've actually managed to find that in a lot of patients who have cystic fibrosis but are actually really mild, they actually have long-term persistent infections of things like Pseudomonas. But even though we can plate them out or molecularly have a look at them and see that they've got all these antibiotic resistance genes, they're not actually causing an infection. So people can actually have really long-term infections. So they're there all the time, but they're not actually doing anything. They're just chilling in the lung, just quite happy. So the problem we have is that when a patient comes in, regardless of whether that that pathogen is infecting or just chilling, we treat it as an infection anyway. And because of that, and because we bombard it with antibiotics, we could be taking perfectly happy bacteria in the lung and causing them to become angry and start actually making an active infection. I hope that makes sense. My brain just had a bit of a brain fart there. But yeah, so there are lots of resistant infections, but they're not necessarily doing anything. They might be there, but they might not actually be bothered about anything, if that makes sense.
Yeah. Well, for me, so we talked about obviously the microbiota. That mainly describes the bacteria. So we call it the microbiota, or the fungi, and the virome is the virus sort of population. In the gut, I mean, I've done a lot of reading around the subject. Obviously, I've yet to see a paper describing the gut myco, as in the fungi, the microbiota in the gut. So for me, if I can get that data, I've got the samples. It's just really tricky to get the DNA library together, because when you work with fungi, there's a lot of contamination, and the little primers we use to amplify the DNA, they're just terrible. They're just not very good. And there's a lot of bias there. So there's so many hoops you've got to jump through before you can, because the last thing you want to do is publish and say, oh, I found this. And then someone goes, well, your PCR primers were terrible, or you've introduced so much bias in your sample that we don't trust you, and then you've tarnished your reputation. So treading lightly, that's the big thing for me, is to actually characterize the gut microbiota, and then see if it actually, if there's any inter-kingdom crosstalk, so bacteria, fungi, is there a relationship between certain fungi and certain bacteria, vice versa? So yeah, that's my main thing. And obviously look at CFTR modulators. Do they actually help or not?
Yeah, mine's basically the same. I'm locked currently in a war with fungus and getting results from samples with fungus. But ideally, I would like to be able to take some samples. I can get the bacteria out, no problem. I can kind of get the fungus out. I've never touched the viruses, but I would love to do a triple threat and do all three on a load of samples and just do that over time and see how they all interact and fluctuate. And yeah, that would be just lovely.
I think one thing to add as well is the work we do, it's quite multidisciplinary. So like, there's elements of our job that I'm really bad at. And there's things that I'm good at. Like, for example, we didn't really touch on it. There's a lot of coding that goes on.
Oh yeah.
So like like using Python, R, other sort of like programming languages to like write code, to do all the analysis, bioinformatics. So there's things that will appeal to someone. There's so many different parts of the job that would stick out to someone and think, oh, I really like that. And that would be enough for you to take it and then make a career out of that and go whichever way you want with it, which I think is an important thing to highlight. But yeah.
There was I visited a lab before where they had a lass who was just finishing up after her GCSEs before she went into college, and she was doing like a summer placement, just helping process samples. So things like that are available. I think universities do like summer schools and stuff in various places. So you can go and get like a little tester of things. The problem is because a lot of this work is kind of really sensitive and you need to be properly trained in some of the techniques. A lot of it will be very basic stuff, but it will give you a kind of flavor of like the workload and what is expected of you, which is very useful.
Yeah.
But yeah, I would.
I think it's definitely the staple techniques like making agar plates or extracting DNA from soil. I don't know, or obviously the samples we work with, there's potential pathogens in there, so you wouldn't want to let that loose on someone who's never been in a lab before. You'd have, we work in really fancy cabinets that are safe and stop biohazards and stuff. But there's definitely the opportunity to learn the core staples in a safe sample to practice with.
There is actually, you can actually get a job as a lab technician with GCSEs. So if you wanted to maybe see about it before you started college and things like that, you could see if a local university or a lab or something was maybe offering like a short-term placement, so you could get some work experience in that way. Cool.